there. So you can use the reference set of the colonies and you can just pick which is the positive colony. And again, you can store it in the master plate. So this way your colonies will be remaining protected, or the genes will remain protected for future use also. And you can identify or you can extract which gene is actually, or which colony is actually containing the gene of your interest. So this is what is called screening of the genomic library. Okay, is there any question from anyone? Actually, the techniques. Okay, as there is millions of base pairs of DNA. See, that is the beauty of molecular biology technology. I already told you the restriction endonuclease will be cutting exactly at the like there is a particular technique. Means the restriction endonuclease will be cutting only where there is a palindromic sequence nearby. Okay, so the palindromic sequence will not be happening randomly throughout the genome. So the restriction endonuclease only be targeting, like there are three types of restriction endonucleases actually, or type 1, type 2 and type 3. So mostly used one is the type 2. So there is a specific count, like exactly after this many bases, the restriction endonuclease will cut. Okay. Like if this is the location of the palindromic sequence, exactly maybe after 20 base pair or 30 base pair or something like that, the the restriction endonuclease will cut. So it is means it will be predefined, means the sequence is already located, you are just exploiting it for your purpose. Okay. So since the DNA is made up of a unique combination, like each gene will be having a different set of combinations. So although there are thousands of means genes, or there are thousands of copies of the DNA, but still the restriction sites will be unique, and exactly the restriction nucleus will be cutting at that location only. Okay. Yes, that part we will come later, how insulin is being prepared. Okay, that is included in your syllabus only. And regarding why you need to bake it, you need to fix it. Okay, like kind of heat fixing is needed, so that the DNA will not be means what we can say, it will not be changing the position, or माने एपट सेपट न हो जाओ, जो जगह अच्छे से जगह फिक्स ହେଇକି ଥାଉ, so to stabilize we are baking it. Okay, like we are trying to fix it. ଯେମିତି ଯଦି ଷ୍ଟେନିଂ କଲାବେଳେ ଦେଖିଥିବ, ଗ୍ରାମ ଷ୍ଟେନିଂ ଯେଉଁମାନେ କରିଥିବ ଯଦି, we usually heat fix the bacteria so that it will remain where it is. କାରଣ ଡାଏ ଯେତେବେଳେ ତମେ ଦଉଛ ବା ପ୍ରୋବ୍ ଯେତେବେଳେ ଆମେ ଯେଉଁ ୟୁଜ୍ କରୁଛେ, ଯଦି ଏଇଟା ମୁଭ୍ କରିବ ବା ମୁଭେବଲ୍ ଥିବ ତାହେଲେ ବାଇଣ୍ଡିଂ କରିବାରେ ପ୍ରୋବ୍ଲେମ୍ ହେବ. So you need to fix it. That is why we are dyeing it. Okay. Sorry, that is why we are baking it. Okay. So next, coming to the thing I was telling you, there is two types of libraries there. One is called the genomic library. The other one is called as the cDNA library. Now, what is the difference between genomic library and cDNA library? Now, since in the genomic library we are conserving the entire set of genome, if it is for a eukaryotic DNA, you are conserving the sequence of intron as well as exon. So where the intron may not be of any use to you. So in order to avoid that part, another library is created which is called as the cDNA library. So cDNA library is the library which is made from the mRNA. So here indirectly what we are doing, we are eliminating the intron part. We are only trying to preserve, or we are only trying to store the exon or the coding part of the genome. Okay. So what are the steps of constructing a cDNA library? You can extract the mRNA. Then using the mRNA as a template, you can construct cDNA, that is the complementary DNA. Then the complementary DNA can be used to form the second copy or the second strand of the DNA, and you can make multiple copies of that DNA, and you can store it in vector and ultimately you can store it in the rest of the bacterial cells or in other type of host cells. Okay. So this is an requirement of the construction of the cDNA library, that is the oligo DT bits, which will be utilized to actually isolate the mRNA with a poly A tail. So in molecular biology, you will be at least you will might be knowing that there is a polyadenylation process is done to a 5 prime capping, and a polyadenylation is done to the matured mRNA in case of eukaryotic organisms for stabilizing the mRNA. So polyadenylation means towards the end of the mRNA, means multiple copies of A. Adenine is added to form a poly A tail. So since the mRNA will be having the poly A tail, if you are creating a vial or if you are creating a column where you have attached DT thymine beads you have added, then ultimately the A will be complementarily bind to the T, and through the poly A tail the mRNA will remain trapped in the column, and the rest of the RNA and the rest of the unrequired material will simply pass through the column. And after the binding, you can use like elution. There is a buffer called as the elution buffer, which will actually release the bound mRNA along with the oligo DT bits from the column to your vial, and that can be used for the construction of the complementary DNA. So how exactly it is done? Let's see the steps. So here, as you have used the oligo DT primer, the complementary tail of the mRNA is bound to the DT primer. Okay. So now this mRNA is trapped here. So you can incubate this mRNA with the reverse transcriptase enzyme. So the reverse transcriptase, what it can do, it can use the mRNA as a template to synthesize a copy of DNA. Okay. So the complementary DNA or cDNA is synthesized in means in opposite of the mRNA strand. So when the cDNA strand is completed, you need to separate the DNA RNA hybrid, because this part of the strand is RNA and the rest part is DNA. So the cDNA, after the completion of the cDNA strand, you need to hydrolyze, or you can simply remove the RNA strand. Now, since it has an open 3' end, it can form a loop kind of structure and it can continue the synthesis in complementary to the existing cDNA. So again, you can incubate with DNA polymerase to synthesize the second DNA strand, and you can use the S1 nuclease, that is another type of nuclease enzyme, to cut the DNA loop. This loop, whatever is found, you can cut it and you can have a double-stranded DNA. So incubate the with terminal transferase to add the single-stranded tails. The double-strand cDNA is ready for insertion into appropriate cloning vector. So what is the advantage over the genomic DNA library here? You don't need to copy or you don't need to carry the introns along with the exons here. So since here you are constructing this DNA from the mRNA, this DNA is actually made up of only the exon sequences. Okay. So same thing that is so here it is actually you are adding the sticky end to make it a sticky end by adding the terminal transferase bases so that it can be easily integrated into any of the cloning vector. Similarly, here also you can synthesize by homopolymeric tailing, means there will be no additional tails over here, but still this double-stranded cDNA can be used in with a blunt end restriction endonuclease to be inserted into a vector. So the process is almost similar. Okay, if there is any question. Yes, this one is biotechnology is going on. Okay, block 4. So this is the terminal transferase plus DCTP. So DCTP means actually this will be carrying the cytosine bases. Okay, so as you can see, the cytosine bases are being added to one of the DNA strand. So this is the RNA DNA hybrid has been constructed. This RNA is now degraded, and this since you have already extended a tail, you have created a strand for to be means you have created a blank strand, or you have created a template strand which can be complementary construct a new DNA strand. So in complementary to CCC, GGGG can be added, and the rest of the DNA can be completed by complementary base pairing. Okay. So there are two ways by which you can construct the double stranded complementary DNA. Then you can insert it into any vector for storage purpose. Next, what we have is hybridization, the screening process that we already saw. Let's see once again the probe I was telling you. So what is a probe, how exactly it is used to screen the gene of your interest. So here you can observe the technique how exactly it is being done. So this is the DNA probe. Probe means this is a sequence of DNA, short fragment of DNA, but you already know what is the sequence of this DNA. Okay, and you have made this DNA probe which has actually almost complementary base pairing with your target DNA sequence. Let's say someone was asking regarding insulin gene. So let's say you know the sequence of insulin gene. So whatever probe you have designed, actually that probe has a DNA sequence which is complementary to the segment of a insulin gene. Okay, so, and again you are tagging this DNA probe with a fluorescence label. Okay, so what is the need of this fluorescence labeling? The fluorescence labeling can help you in easy detection. So if you remember in the last class we discussed that CMA, the microarray technique we were discussing. So there you could observe that there were different color products were there, green, red and yellow colors were being emitted. So that is the utility of fluorescence. Okay. So next you can denaturate the probe, because since it need to bind to your target DNA, it must be single stranded. So this probe will now go and find the target sequence, and it will form a complementary binding there. So the complementary binding will lead to release of fluorescence emission, from which you can easily detect which colony of the bacteria or where actually your gene of interest is located. The rest of the colonies that do not have the target DNA sequence complementary to your probe will not have this kind of fluorescence emission. Only the colonies that will have the gene of your interest and where the complementary binding will happen can only give you the fluorescence emission. From there, it will be easy for you to screen the gene of your interest.
Ma'am, is there any possible to the probe bind to any other segment of DNA or any virus DNA?
Yes, sometimes it might happen, which we call as the non-specific binding, but that is very rare because let me tell you a very interesting thing. Actually, if you look into a segment of DNA, okay, so DNA is made up of word sequences like A, T, G, C, like this, right? CG and so on. So if you look into this pair AT, okay, or this sequence, you might find that within the genome the AT might be repeated like 1,000 or 1 lakh times. Now, if you consider ATG like this, so this sequence also might be repeated many times. Like that, if you consider a sequence of, let's say, around 20 base pair or something, that sequence repetition will not be found more frequently within the genome. You understood my point? So that
Yes,
is
ma'am.
why most of the probe that are designed, they will be of a definite length. That length repetition in the genome is very rare. So it will not be binding to any non-specific location. Okay. So the binding becomes very, very specific. So these are actually very meticulous jobs which are done with the help of bioinformatics. So it's not like they can go and bind somewhere else, but sometimes it may happen, and there you may get a false positive result, but that is very rare. Usually the binding is very specific, because as I told you, the sequence that is designed or that is targeted, it will not be occurring in the genome anywhere else. It will be very rare.
The
Just like we
probability
had,
will be less.
A probability might, there might be, sometimes it might occur, but it is very, very less. Okay. Like our
Madam,
fingerprinting. Okay. So it is something like that. It will be very, very unique. The sequence will be very unique. Okay.
madam,
Yes,
what
any
is
other?
the function of probe?
Probe, I told you, probe is actually you are using the probe to see. For example, you say there are thousands of logs are lying there. Okay, means ମାନେ ବହୁତ ସାରା ତମେ ଗୋଟେ ରୁମ ରେ ତାଲା ପକେଇଛ. Okay, now the target sequence is like that. Let's imagine the target sequence is ମାନେ ଆମେ ଯୋଉ ତାଲା ଗୁଡା ପକେଇ ରଖିଛେ, the target sequence is that. So each ତାଲାର ଗୋଟେ ଚାବି ଅଛି କି ନାହିଁ? each ତାଲାର ଗୋଟେ key ଥିବ. So that key is the probe. ତମେ ଜାଣିବ କେମିତି ଯେ ତମର ଯୋଉ interest of ମାନେ ଯୋଉ ବି lock ଟା ତମର interest ଅଛି ସେଟା କୋଉଠି ଅଛି. So you will be using the probe as a key to find out the lock. Okay, so this is actually a complementary sequence. The probe is having a complementary sequence to the target DNA sequence. So ତମେ ସେ lock ଯୋଉ ଅଛି ଆମର different ଯୋଉ key ଥିବ ସେ key ଗୁଡା ସବୁ lock ରେ ତ ଯିବନି ନା. It will be fitting to only the lock which is complementary to that key. So the probe is like that key, and moreover you have tagged that key. Okay, so माने तमे maybe तादरे किसी ओटे ring लगे दीचो, so that you can easily identify कि एक key टा जे कि जो lock रे लागिबो, that lock is my requirement. Okay, so it is something like that. This is your target DNA sequence. तमे जो actually जेताले तमे जो library रे store करिक रखुचो ना, so you don't know exactly which segment of DNA is stored in which library, but you know the gene and you know its sequence. So तमे कौन करछो एक probe design करछो जो probe टा के complementary sequence to your target DNA. So आउ तमे कौन करछो अपने probe रे उटे fluorescence tag करछो. So जो जगह रे ये probe complementary DNA टा पाइबो ये जीवो ता सहित bind करिवो जे केमती bind करछि देखो एठी. Okay, and bind करके गोटे fluorescence emission दबो. So से emission रो तमे जानि परिबो कि this colony is having the gene of my interest. एबे बुझ पालो जेमती मो प्रीवियस गोटे स्लाइड देखेथिला जो ऑटो रेडियोग्राफ वाला ए जगह रे. ओके, सो एठी हमें यूज करथिले फॉस्फेट यूज करथिले जो रेडियो लेवल फॉस्फेट, but instead of radio level phosphate you can also use a probe. Probe is nothing but a short sequence of DNA with a known DNA sequence and attached to a fluorescence tag, fluorescence dye. So ebe बुझिलो कि आउ कार किछि क्वेश्चंस अछि, you can ask. Okay, so next we have some techniques like your southern blotting, northern blotting, etc. So let me briefly explain this. What is this technique exactly? So southern blotting is like it is the similar like it is a technique used to identify the same, that is the gene of your interest. So basically you are identifying the DNA or the segment of DNA which is carrying the gene of your interest. Okay. So you have your genomic DNA. So I told you, since the genomic DNA is so large in size, you cannot keep them in a single organism, or you cannot put all that genomic DNA into a single vector. So what you will do, you will try to make fractions of this. Okay, small, you will try to cut it into small pieces. So the small pieces are created by the restriction endonuclease. Then, since they are made up of different sizes, you can simply run them in the agar or gel to separate them on the basis of their size. Okay. So the smaller fragment will travel towards the end and the larger fragments of DNA will remain in the upper part. So the high molecular weight of DNA will be remaining in the upper side. The low molecular DNA will run to the lower side. Okay. Now, what you can do, you can transfer this agar or gel into a blotting paper. So the paper is called as nitrocellulose or a nylon filter paper. Now, since this agar or gel is containing all the genomic DNA or the fragments or the genomic DNA in different fragments. Now, all the fragments are not of your interest. You need only one fragment that is of your interest. So what you will do? You will use a probe, tagging it with a phosphate that is radioactive, P32. Okay, this is the radiolabel phosphate. So, phosphate is actually a part of your DNA nucleotide chain. Okay, so what this probe will do? It will go and it will bind to the segment of DNA that will have complementary DNA sequence to this probe. So see, there are a lot of DNA strands are there, but does it bind to all the strands? It's not binding. So where it is binding, it is only binding to the segment of DNA that has a complementary base pair to this probe. And when you will carry out the autoradiogram or you will expose this to the X-ray film, only this part you can see that it is illuminated or it will glow like that. Okay, so you get to know that this part of your DNA or this segment of your DNA is containing the gene of your interest. Okay, so this technique is called as your southern blotting. You understood this part or not? Actually ଟିକେ ଯଦି ଲାବୋରେଟୋରୀ ଆଇଡିଆ ନଥିବ ଟିକେ ମେ ବି ଡିଫିକଲ୍ଟ ହେଇପାରେ, ସୋ ମୁଁ ସମସ୍ତଙ୍କୁ ସଜେଷ୍ଟ କରିବି କି ଏଇସବୁ ଜିନିଷର ବହୁତ ଗୁଡ଼ାଏ ଭଲ ଭିଡ଼ିଓସ୍ ଥାଏ, ମାନେ ଏକ୍ଜାକ୍ଟଲି କେମିତି ଲ୍ୟାବ୍ରେ ଟେକ୍ନିକଟା ହେଉଛି ତୁମେ ଦେଖିପାରିବ। ସୋ ସମସ୍ତେ କିଛି ନା ୟୁଟ୍ୟୁବରେ ଜଷ୍ଟ ଏମିତି ସଦର୍ନ ବ୍ଲଟିଂ ଏମିତି ହିଁ ମାନେ टाइप करदेले तुमको बहुत गुड़ा इनफॉर्मेटिव वीडियो मिल किंबा आ गोटे भी करिपरिबो जेते हमर जो गुड़ा सब इक्विपमेंट सप्लायर्स थांती जेमती गोटे थर्मोफिशर साइंटिफिक जेमती हला हाई मीडिया सो एमानकर भी बायोरेड एमानकर भी सब माने वेब पेज रे एमानकर भी सब टेक्निक गुड़ाकर बहुत गुड़ा वीडियोस थाए। सो जो मान को टिके डिफिकल्ट होची त माने वीडियो को रेफर करि परिबो बुझिया पे टिके इजी हबो। एटा सब लैबोरेटरी बेस्ड जिनस सो जो मानकर लैब एक्सपोजर थिबो से मानको टिके कैच अप करिया पे इजी हुए और एल्स इट विल बी लिटिल बिट डिफिकल्ट टू कोरिलेट जेहेतु तुमे माने प्रैक्टिकली जिनसटा देखिनो यू माइट बी थिंकिंग एटा केमती होथबो बट दिस एक्चुअली हैपेंस इट इज वेरी वेरी प्रिसाइज एंड इट इज वेरी वेरी स्पेसिफिक।
Ma'am, sometimes radio level probe is used and sometimes fluorescent probe
Fluorescent
is used.
stat probe is used. Yes, you can use
That
as
means
per your convenience. Okay. See,
Ma'am,
for the
can we?
blotting technique, actually, for the blotting technique, usually this radio level probe is used. Okay. And when you are screening like through PCR or something, then you are using the fluorescent stat. Okay. Okay.
Ma'am,
Any other question?
can we use exonuclease on the place of endonuclease
No,
by
no,
cutting DNA?
exonuclease will not be any useful because exonuclease means it will cut DNA at one of the ends. But in the genome, okay, you have means the genes will be located inside the genome, not at any part, any end. And the exonuclease will only cut at the terminal position. Exo means outside. Okay, so you have
Yes,
to
yes.
use the endonuclease only. Okay, so that it can cut anywhere in the middle portion of the DNA like this.
Okay.
Ma'am, every gene has their unique restriction site.
Yes, every gene will have their own unique restriction sites. Okay. No, the
Ma'am,
restriction,
which
see, the restriction sequence for a means enzyme is fixed, but every gene will have its own restriction site at different locations. Okay, like the EcoRI will only recognize the palindromic sequence is fixed, but each gene will have its own set of palindromic sequence. Okay, so that is why when this restriction endonuclease is subjected, it will cut into multiple pieces.
Can the restriction site at the middle of the gene?
Usually no, they will be towards like the terminal part, because if it will be in the middle of the gene, then what will happen? The function of the gene will be disrupted. Okay, but sometimes what happens when the mutations will be there, then there will arise a restriction site in the middle of the gene. I don't know if you remember or not. I showed you one slide in the sickle cell anemia. condition. There, if you remember that diagram, let me draw that for you only. Okay, if you remember,
Yeah.
there was a graph like this. For a normal gene, you were getting a single band like this. Okay, for a heterozygous, there was a band like this. And for the sickle cell person or for a sickle cell DNA, the band was like this. So this is actually nothing but the restriction map of the gene. So usually, this HBB gene has two restriction sites. Okay, like this. So the two restriction nucleus will cut here, right? So how many bands you will get? Let's say this is of 13 kb. So both the chromosome or both the copies of the gene will produce a segment which is 13 kb. So this part, this one. So when you will run it in the agar gel, only one segment you will get. This is the 13 kb unit. Okay. Now when there will be a mutation, what will happen? There will be a second restriction site arises here. Mutation means change in the DNA sequence. So maybe that new sequence will be a restriction site for a new restriction endonuclease. So now this, if there is a heterozygous that is having a normal gene and there is a a mutated gene is there. So in that case, in case of heterozygote, this gene will be cut by only the restriction endonuclease in both sides. So you will have a 13 kb segment, and this can be cut at three different locations. Okay, so you will have a band that is 11 kb and 200 kb. Okay, so you will have a band like this, which is this band like this one you will be getting. This is the normal one, and here alongside this you will be having two different bands like this. Okay, 11 kb and 200 kb like this. And if there is a and if there is a means it is a homozygous recessive, means it is a sickle cell person. So you will have two bands like this, 11 kb and one 200 kb. Okay, this will not be, yes, it will be 13 kb, 11 kb and 200 kb. Here it will be like this, 11 kb and 200 kb like this. So sometimes due to mutation, a new restriction site may arise.
And how many base pair the restriction enzyme can identify?
It depends actually. The palindromic sequences are different for different set of enzymes. You have to means memorize that part. Here I have cited I think only 2 to 3 examples. Like here you see, this is visible to you. Okay, so this is this EcoRI has a recognition sequence of 6 nucleotide bases, 6 base pairs, 1, 2, 3, 4, 5, 6. But this BAM HI, it has a sequence for 1, 2, 3, 4, 5, 6. Again 6 base pair. But this ALU one, it has a recognition sequence which is only 4 base pair. AGCT, AGCT. Okay. Similarly, and look at the cutting position also. So here it is creating a sticky end. Sticky end means it is unequal. Okay, so here the ligation is little bit easier because a similar cut will be made on the vector also. So the vector will have a complementary pairing to this part of the gene, and they can easily ligate. Okay, but here it is creating a blunt end. See, these are also segment of DNA. So you need to have complementary bases to complete the ligation process. But here it is the blunt end. So here you have to carry out certain modifications to make it sticky so that the ligation can occur. So what I mean by the ligation is, just look at here. There is one diagram here. This one. Okay. This is visible to you.
Yes,
See,
ma'am.
yes. See, this is the equari which has created a sticky end like this. Okay, so since this is sticky, let's say this is G. Okay, the upper part is G and the lower part has nothing. So similarly, if you have another cut which has a C, the C can come and pair here. So you can have a ligation product like this. Okay. So that is the utility of a sticky end. But if it is a blunt end, so blunt end means it like the other DNA fragment cannot have a complementary binding here. So you have to convert this blunt end into a sticky end. Then after you form a sticky end, the ligation can be completed. Okay.
And the restriction side is known by any trial and error method, or there is any other method?
No, no, there is specificity only. It is prefixed. The restriction endonuclease that means the EQRI or the BAM-HI, the palindromic sequence is fixed for them. So if only the sequence is like that, then only they will cut.
And
It's not like
we should have prior knowledge of DNA sequencing to restrict the DNA.
you must know that like what is the palindromic sequence for this DNA you need to know, or else you cannot use that particular restriction in enzyme for that sequence. See, actually in
Madam,
the DNA all the sequences will be present. Okay, most of the sequences will be present. So depending upon which restriction enzyme you are using, accordingly the DNA will be cut. Actually, I don't know if I could answer your question or not. Can you please repeat your question? What was your query exactly about?
as a cord DNA sequence is 5 days to 3 days.
5-3. No, it will cut in both the sides. It will cut in the 5 prime end also. It will cut in the opposite end also. See, here you can see. I
Okay.
showed you one diagram there, how exactly the restriction endonuclease is acting. Here, see, the site of cleavage is actually fixed. Here you can see. Okay. So where it is cutting in the 5 prime end, it is cutting after the G. Okay. So you have one segment that is 5 prime G, and the other segment is 3 prime C T T A A. Similarly, from this side, the cut is here after the G. The site of cut is actually exactly the same for both the strands. So it will cut where? After the G in the 5 prime sequence. So here again you have the means separated strand, that is G, 5 prime G is here, AA, TT, C, 3 prime is here. So in the vector, what will be the strand? In the vector, the strand will be similar to this. So what will happen? This 3 prime CTTAA will be from the vector that will come and complementarily bind to this. Similarly, this G part will be complementary in the vector that will come and bind here, so that the restriction side can be completed. Okay, so in the vector segment also, one fragment will have this DNA, the other fragment will have this DNA, so they will be complementary binding to each other. See, look at here. Okay, this is
Means,
how the
yeah,
sticky end is created. So this is, let's say, for this strand, so the vector will be exactly complementary to this. So now the DNA strand can be completed. Okay. So it will be like a linear DNA again, or circular DNA if it is inside the bacterial genome. Okay. Okay. Any other question?
no, madam.
Okay, fine. So let's proceed. So, like we are doing the southern blotting for the DNA to identify or to find out the DNA of our interest. The northern blotting is done for the RNA. Okay. Similarly, there is a western blotting also, which is done for the protein. Quantitative, qualitative analysis of proteins. Sorry. So remember the three names: southern is for DNA, northern is for uh, RNA, and western blotting is for protein. So northern blotting technique is used to monitor the gene expression. That is why we are targeting RNA instead of DNA. The sample RNA from the target organism is isolated and separated using the electrophoresis on a formamide agar gel, followed by transferring the isolated RNA to a support membrane. The membrane is usually a nitrocellulose membrane. In the presence of a specific buffer, this can be accomplished using the capillary transfer technique. The membrane is next either baked at a high temperature or subjected to UV cross-linking, which causes the covalent bonding of RNA to the membrane. So someone was asking why we are baking it. So baking, jemti call it is used for fixing, so that it will not move from that place to another place. So either you can bake it at high temperature, or you can simply expose it to UV light, so that it will remain fixed at that location. Okay? And prevents the nucleic acid from being washed away during the later processing. So see, this is the transfer buffer, this is the support sponge, and you have put the gel here. Okay, and this is the membrane, the nitrocellulose membrane. So capillary action means, see, all of you have seen the tissue paper, right? So when you man जेतବେଳେ ओदा surface अछि suppose को जगह रे तमे tissue paper टा पकी दियो, कोन हबो संगे संगे से tissue paper पानी टा soak करि नबो ना. So exactly तमरो nitrocellulose membrane जो अछि सेमती काम करे. सो जो तले जो तमरो जेल अछि जोथरे तमरो आरएनए अछि आइसोलेट हेकना आरएनए बैंड गुडा अछि, तापरे तमे जो नाइट्रोसेलुलोज मेम्ब्रेन टा पकेबो, जेमती कैपिलरी एक्शन रे तमरो हमरो टिश्यू पेपर टा पकी देले तळु पानी टा सोक हेकी पळे आछी ऊपर को, इन ए सिमिलर फैशन तमरो जेल रु ए पेपर को ट्रांसफर हिजबो आरएनए बैंड्स ना. Then you can subject it to further processing. So further processing, there are the steps. Like the RNA molecule is run on a denaturing agar gel. The gel electrophoresis separate the fragments. Staining with the ethidium bromide or ETBR allows the visualization of the RNA molecule. The blotting transfers the RNA band to a nitrocellulose filter or membrane. The gel and the filter allow the transfer buffer to pass through and reach the paper towel. This results in a nitrocellulose filter with the RNA fragments precisely transferred from the gel. A probe for a particular gene that has been radioactively labeled is exposed to the filter. In case the transcript is produced, the probe will bind to the RNA. So the filter is exposed to X-ray film for autoradiography. A band on the film identifies the fragment containing the desired gene. So since I am RNA ko target koruche, that it means that the gene must be expressed. Jodi ote gene express houthubo but transcript कहले हमर mRNA. जदि जो DNA टा तमरो transcribe हे कि mRNA form करिथिबो, then only the probe can go and bind to the complementary RNA strand. So similarly तमरो ऑटो रेडियोग्राफ जनरेट होबो जेमते हमर DNA रे हितला, it is similar to that. And you can use a marker to identify what is the size of the particular gene. Okay, and this is the denatured agar gel. So denatured agar gel कहले कोन ना से सेम RNA टा को different fragment रे fragment कराइछि. देखो ए जेमते अछि ना RNA of known sizes for the markers. Then this is the RNA binding membrane, this is the agar gel solution, and you are using the blotting paper to transfer the RNA from the gel to the paper. So RNA is transferred to the RNA binding membrane. Then you need to hybridize the membrane with the probe. So the probe is actually complementary to the transcript or the RNA of your interest, and you are exposing the filter to a X-ray film. So X-ray film रे जेतु तमे हाइब्रिडाइज करछ रेडियोएक्टिव न्यूक्लिक एसिड प्रोब रे से फॉस्फेट, लेट से फॉस्फेट रे ही फॉस्फेट P32 रे लेबल करछ, जो जगह रे तमरो बाइंडिंग हेतिबो से जगह रे यू कैन ऑब्जर्व ए रेडियोग्राम और ए कलर प्रोडक्ट और ए माने ग्लो यू कैन ऑब्जर्व. So that is indicating that this is actually containing the RNA of your interest. Then the same colony hybridization is there to identify which part of the or which colony is actually containing the gene or the RNA of your interest. See same jinsa, so I am explain karuni. Next, let's go to immunotechniques. Okay. So आमर जेतले कोविड हेतिला किछि दिन पर हमर ओटे किट डेवलप हेतिला to know whether a person is infected with COVID or not, to know whether a person is infected with COVID or not. So all these detection kits are actually based on immuno blotting or immunotechnique. Okay, so there are different types of immunotechniques. One is called as the ELISA, enzyme linked immunosorbent assay. So kali hi ki pacharuthili important question, so this could be a question for your end term examination. Explain the techniques of ELISA, the types and the technique of ELISA. Okay, this could be a long question, 10-mark long question. So there are different, so the principle of ELISA is very simple. It is based on antigen antibody binding. I hope ସମସ୍ତେ ଜାଣିଛନ୍ତି antigen ଆଉ antibody କ'ଣ?
Yes, ma'am.
So antigen is actually any foreign substance, like if we are saying it is a virus or bacteria or something, तो यार membrane ऊपर जो protein अच्छी कि lipopolysaccharide अच्छी किसी भी अच्छी, that can trigger a immune response is known as a antigen. तो ये जो एंटीजन को कॉम्प्लीमेंटरी जो प्रोटीन आम बॉडी प्रोड्यूस करुच्ची वाई शेप रो स्ट्रक्चर गोटे मॉलिक्यूल दैट इज द एंटीबॉडी. सो जो एंटीजन और एंटीबॉडी रो बाइंडिंग स्पेसिफिसिटी बाइंडिंग इज ऑल अबाउट द ELISA और एनी काइंड ऑफ इम्यूनोटेक्निक assay. So we have 4 different type of ELISA. Okay, এটা ফার্স্ট মনে রাখো এলাইজার যে আমার মানে অ্যাব্রিভিয়েশন আছে দ্য এনজাইম লিঙ্কড ইমিউনোসরবেন্ট assay, ELISA, ELISA. So there are different types of ELISA techniques are there. Broadly 4 types are discussed: direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. So let's see को type रो ELISA रे कौन हुए. So direct ELISA is very simple. इतरे कौन करा होची ना, you are taking an or you are coating a microtiter plate with the antigen, or you are coating this plate with a antigen. Now that antigen will be having a antibody. Okay, complementary antibody. So each antigen will be having its own set of antibody. So again you will use the antibody which is conjugated with an enzyme. So this is enzyme linked assay. Okay, so if the antigen or the antibody that you are using will be complementary to the antigen, it will go and bind. Then you will add a substrate. The substrate means a compound that will change color due to the activity of this enzyme which is linked on the antibody. So the color change will actually let you know the concentration of the antigen. If the color produced is more, means more amount of antigen is present, and you can say that the test is positive. If there will not be any color change, means the antibody has not bound to the antigen, or the antigen you are suspecting is not present in the solution. Therefore, there will be no color and we can say the test is negative. So, जेहेतु आम एठी directly enzyme linked antibody use करुचे, we are calling it as direct ELISA. Now, what is indirect ELISA? In indirect ELISA, the process is similar, but there is an extra antibody is used. So the antigen is the well is coated with the antigen, and you are using the complementary antibody to bind the antigen. Next, you are using a secondary antibody. So the antibody that is directly binding to the antigen is called as a primary antigen. Then you are using a secondary antibody that is linked to the enzyme. So jodi tomar primary antibody bind korithiba antigen re, then only the secondary enzyme can go and bind to the primary antibody, and after adding the substrate you will get the color product. So the intensity of the color will decide the concentration of the antigen. जदि बहुत antigen थिबो तमरो माने well रे ताले बहुत गुडा antibody जे bind हबो जते primary antibody bind हबो सेते secondary antibody ता सहित bind हबो color कोन हबो सेते बेसी देखा जिवो. The color intensity will be more. जदि antigen रो concentration low थिबो बहुत low अछि ताले antibody बेसी bind करिवोनी the color product the intensity of the color will be low. So this is a qualitative analysis, quantitative as well as qualitative, means it can give you, sorry, this is a quantitative analysis, means you can actually predict the concentration of the antigen by looking into the intensity of the color product. So this is direct, then indirect. Next we have sandwich ELISA. So sandwich, as the name suggests, here instead ago ditare kon dekhile na, first the antigen was being coated in the well. Now first you will coat the well with antibody. Now washing
And
it
the
up,
same technique is used in malaria kit or pregnancy kit.
yes, the same technique is used, but there the antigen and antibody are different. Here it is actually for the disease point of view I am speaking, but they are also same. See, antigen means actually any kind of proteinaceous molecule, and antibody, that is complementarily will bind to the antigen. The same thing is used in any kind of detection kit, including your pregnancy kit. Okay. So in pregnancy kit, we are targeting HCG, human chorionic gonadotropin hormone, which is a mandatory hormone released during the pregnancy. The same
In
and the
sandwich
technique is also same.
ELISA,
Yes.
in sandwich ELISA, which type of antibody is used?
No. See, the antibody is actually like whatever you are trying to detect. Suppose you are trying to detect HIV. Okay. So you know the HIV antigen. What is the HIV antigen? You know. So accordingly, you will be utilizing the antibody of HIV. So instead of antigen, first you are coating the well with antibody. Then washing is done to prevent, means to release any kind of unbound antibody. So जदि किछि bind है नथिबा सेटा wash रे release हे जिवो. केवल जति कि well रे bound हेबे सेही तक antibody रहिबो. Then next you are adding antigen to it. So जदि तमरो specific antigen थिबो जिवो antibody रे bind करिवो or else bind करिवो. Then you will provide it with secondary antibody linked with the enzyme. So same thing, जदि तमरो primary antibody रो complementary antigen अच्छी और bind करिछि, then only it will have space for binding of the secondary antibody. So secondary antibody bind कला परे तमे product reactant देलो, then reactant will be converted into a chromogenic product, जो through तमे color or intensity measure करके you can predict what could be the possible concentration of the antibody in the sample solution. Yeah. The only difference is तमे direct और indirect ELISA रे antigen आगे use करूଛ, but sandwich ELISA रे you are using antibody instead of antigen. तापर आसीबो competitive ELISA, एटा टिके slightly different from the rest of the three. So competitive ELISA रे कोन हुए, you are using the antibody along with antigen bound antibody. So दी प्रकार antibody तमे use करूछ. Like for example, let's say you want to find out a person with your what we can say hepatitis virus. Yeah. So you want to find out whether a person you are suspecting is having hepatitis or not. So से जगह तमे कौन कलो ना hepatitis antigen bound antibody plus खाली antibody use कलो. तापरे well रे कौन कलो hepatitis antigen कोड कर देला. तापरे याकू ऐड कलो. अच्छा एबे जदि तमरो ए सॉल्यूशन रे हेपाटाइटिस बी रो concentration बहुत अधिक थिबो ताहले कोन हबो, मोस्ट ऑफ द एंटीबॉडी कोन हेतिबे ऑलरेडी बाउंड हे कि थिबे. ओके, ताहले एठी कोन हबो ना बाइंडिंग ता कम हबो, कारण एठी ए एंटीजन सहित तमर के बाइंड करिवो ना जो एंटीबॉडी रे एंटीजन बाइंड हे कि नाही. तापरे सिमिलर टू द प्रीवियस केस या कोटे सेकेंडरी एंटीबॉडी देलो एंजाइम लिंक्ड, reactant देला product को measure करिवो. So एठी कौन हबो ना it will be inverse माने जदि product बहुत अच्छी सॉरी जदि तमर आमर कोन ए जो रेडियोएक्टिव बा कलर इंटेंसिटी जदि बेसी अच्छी ता माने तमर ओरिजिनल सॉल्यूशन रे एंटीबॉडी सॉरी एंटीजन कम थिला बट जदि एठी कलर इंटेंसिटी कम अच्छी मीन्स तम ओरिजिनल सॉल्यूशन रे एंटीजन रो concentration अधिक थिला. ओके सो आई एम रिपीटिंग दिस वंस अगेन. आगुरु आमे कोन करथिले ना खाली एंटीजन नले एंटीबॉडी यूज करथिले बट एठी आमे कोन करछे ना वी आर इनक्यूबेटिंग द एंटीजन विथ एंटीबॉडी along with some free antigen. So एठी सपोज तमे जो सॉल्यूशन नेचर सपोज हेपाटाइटिस बी रो गोटे सॉल्यूशन नेचर बा गोटे पर्सन रो माने सेरम आइसोलेट करिचो जाहा को तमे सस्पेक्ट करचो कि ताको हेपाटाइटिस बी हेची. सो हेपाटाइटिस बी हेची माने कोन थिबो तारो हेपाटाइटिस एंटीजन बहुत थिबो एचबी एंटीजन जदि मु कहबी ताको कोन बहुत अछि. सो जदि एचबी एंटीजन बहुत अछि तमे जेतले ई सॉल्यूशन को एंटीबॉडी रे मिसेबो ताले कोन हबो मोस्ट ऑफ द एंटीबॉडी विल बी ऑक्युपाइड बाय द हेपाटाइटिस एंटीजन आ फ्री एंटीबॉडी बहुत कम थिबे. तापरे ए मिक्सचर को दियो गोटे एंटीजन कोटेड वेल रे. अच्छा ये एंटीजन कोटेड वेल रे ए जो एंटीजन अच्छी वाल रे जो एंटीजन कोट हिची ता सहित किए बाइंड करि परिबो ए बाउंड एंटीजन बाइंड करिवो ना फ्री एंटीबॉडी बाउंड करिवो. फ्री एंटीबॉडी जे अछि सी बाइंड करिवो. सो तमे फ्री एंटीबॉडी बाइंड करि गला तापरे पुनी सेकेंडरी एंटीबॉडी देला तापरे कलर माने क्रोमोजेनिक सब्सटेंस देके कलर मेजर कलो. सो जदि तमरो फ्री एंटीबॉडी बेसी थिबो ताहले कलर कोन बहुत आसीबो. सो फ्री एंटीबॉडी बेसी अछि माने कोन हिची ना इठी तमरो हेपाटाइटिस एंटीजन कम थिला सेथि पई कि मैक्सिमम एंटीबॉडी आर फ्री आ तमरो से माने एम्ति प्रोसेस रे बाइंड करके बेसी कलर इंटेंसिटी दोचंती. जदि तमरो ओरिजिनल सॉल्यूशन रे हेपाटाइटिस बी रो कंसंट्रेशन बहुत अधिक अछि ताले मोस्ट ऑफ द एंटीबॉडी विल बी ऑक्युपाइड आ ऑक्युपाइड एंटीबॉडी गुडा एठी कोन बाइंड करि परिबेनी ए एंटीजन रे बाइंड करि परिबेनी ताहले तमरो कोन कलर इंटेंसिटी विल बी लो. So a previous case हमें कौन देखुथिले ना more is the color more is the concentration of antigen or antibody किंतु competitive रे कोन it is reverse. The more is the color intensity means less is the antigen concentration, the less is the color intensity means more is the antigen concentration in your sample solution.
And why the second type of antibody is attached to the secondary antibody?
Sorry, why?
The second type of antigen is attached to secondary antibody.
Where? Wait. This figure you are asking?
Competitive ELISA. There is two type of antibody. One is free and one is attached to any kind of antigen.
No, not any kind of antigen. This is antigen of your interest. Okay. So both the antibody are actually, they are complementary to your antigen. But depending upon the concentration. Suppose तुम्हारे 100 टा antigen अछि. तमे 150 antibody use करूछ. ताले केतेटा एंटीबॉडी फ्री रहला 50 टा फ्री रहला ना से फ्री एंटीबॉडी गुडा इठी जाई बाइंड करिवे बट जदी
there is already antigen is coated in the ELISA well
no that is coated. See here you are actually allowing a competition between the existing antibody and the antigen that you are already suspecting with the experimental antigen to find out in what concentration it is present. So that is why we are already using an antigen which is coated to the antibody. In the previous case, we are simply using the antigen or antibody to find out their concentration. Here, in the sample solution, you know that it might be containing some of the antigen, but still you want to use like in what concentration it is present, whether it can compete with this this antigen that is you are using experimentally to replace it or not. So that is why the name is given as competitive ELISA.
Ma'am, in the third case, there is no antigen attached to the enzyme-linked antibody. So why
Here,
we need the
here,
antibody like this?
this one are you asking?
Yes, yes, ma'am.
Sorry, I did not understand your question. What is your question?
In this figure, there is no antigen attached to the enzyme-linked antibody.
No, no, antibody will never be, sorry, antigen will never be directly attached to the enzyme linked antibody in this case. Here, this
Just
antibody
the
is
prevent
complementary
the
to the primary antibody. Okay. So, see, what is happening is here, this is the infected antigen that you are using from the patient sample. Okay. And this is the antigen that you are using, which is means from outside, like you are experimentally using this antigen. So see, only the antibody which is free will go and bind to this wall coated antigen. Okay, and this antigen will not be, this antibody which is already having the antigen will not be bound to this one. So this will be washed in the next step. So only the primary which is bound to the antigen, there only this enzyme linked secondary antibody will go and bind, and it will give the color product like this. So this only the free antibody from this solution that you are using will bind to this antigen. Okay. So the more is the amount of free antibody means the more will be the color. So if the color is more means in your original sample, this antigen concentration is less. That is why you have more free antibody which can go and bind here. But if in the original sample, your antigen will be more, means this disease antigen will be more, then there will be very few antibody left. So they will not go and bind, or the binding will be less in this case. So the color generated will be lesser.
Ma'am, the fast antibody is fluorescently tagged. So we
No,
can.
no, no. The fast antibody is simply, which one you are asking? This one.
Ha, yes, ma'am.
না না এটা শুଣ এটা actually দেখো এই কেসরে তুমি ডাইরেক্টলি অননোন স্যাম্পল ইউজ করুছ ହେଲା তো আইদর তুমি ডাইরেক্ট আন্টিজেন ইউজ করুছ কি আন্টিবডি ইউজ করছ প্রিভিয়াস কেসরে বট जो কম্পিটিটিভ এলাইজা কেসরে তুমি কোন করছ ইউ আর অলরেডি ইউজিং এ মিক্সচর অফ আন্টিজেন এন্ড আন্টিবডি ହେଲା সো তুমি সপোজ যেমতি কইলি কি হেপাটাইটিস সপোজ ইউ ওয়ান্ট টু ডিটেক্ট কি হেপাটাইটিস গোটে লোককু হইছি কি নাই सो तमें तार जो सैंपल बाहर कर हेपाटाइटिस एंटीजेन ऑलरेडी तमें हेपाटाइटिस एंटीबॉडी भी देछ सो जदि हेपाटाइटिस एंटीजेन से लोकर बहुत बेसी थिबो इन्फेक्शन लोड जदि बहुत अधिक थिबो ताहले से एमती ऑक्युपाई हे कि थिबो ना तार फ्री एंटीबॉडी बहुत कम रहिबो सो जेतले तमें याकू पुणी एंटीजेन कोटेड वाल रे दबो से फ्री एंटीजेन केवल जाय कि बाइंड करिवो ना इठी आउ ये भी कम्पीट करिवो बाइंड करिवा पई कि किंतु जदि यार माने कंसंट्रेशन बेसी थिबो ये कम्पीट करी एठी बेसी बाइंड करि परिबेनी कारण यार कंसंट्रेशन कम अछि सो सेही जगह रे कोन होबो तमर केवल फ्री एंटीबॉडी गुडा जाय कि बाइंड करिवो सो जेहेतु ऑलरेडी तमर यार कंसंट्रेशन बेसी अछि जो हेपाटाइटिस बी रो माने हेपाटाइटिस एंटीजन रो कंसंट्रेशन बेसी अछि सो एई जगह रे तमर फ्री एंटीबॉडी बाइंडिंग बहुत कम होबो सो बहुत कम जदि हबो कलर प्रोडक्ट विल बी लेस एबे बुझ पालो ना बुझ पारनो
Yes, ma'am.
पुणी नेक्स्ट के कोटे कंडीशन कोन है देखो एडी द ओनली डिफरेंस इज ए जो प्रीवियस केस रे हमें आइदर एंटीजन की एंटीबॉडी खाली डायरेक्ट यूज करथिले बट एठी कोन करछे हमें जो सॉल्यूशन टा दोचे इट इज ऑलरेडी ए मिक्सचर ऑफ एंटीजन एंड एंटीबॉडी एंड ऑलरेडी फ्यू एंटीजेन्स आर बाउंड टू द एंटीबॉडी हैला सो एठी एंटीजन रो कंसंट्रेशन केते अछि दैट विल डिटरमिन कि तमरो एई जगह रे जो माने फ्लोरोसेंस एक्टिविटी केमती हबो बाय जो कलर इंटेंसिटी कोन हबो जदि तुम ओरिजिनल सॉल्यूशन रे ऑलरेडी एंटीजन कंसंट्रेशन बहुत अछि ताले सब एंटीबॉडी गुडा ऑलमोस्ट ऑक्युपाई हे कि थिबे सो फ्री एंटीबॉडी बेसी नथिबे ताले कम फ्री एंटीबॉडी गुडा जाय के कोट वाला एंटीजन रे बाइंड करिवे तापर सेमते सेकेंडरी एंटीबॉडी बाइंड कला आ जेमते कलर फॉर्मेशन होतला सेमते फॉर्म होला बट जदि एठी यार कंसंट्रेशन बहुत अधिक अछि माने तमरो फ्री एंटीबॉडी कंसंट्रेशन बहुत कम अछि सो कलर प्रोडक्ट कोन हैला लेस हैला जदि तुम एठी ही एंटीजन कम अच्छी ताले बेसी गुडा एंटीबॉडी कौन रही फ्री रही सो जितेक फ्री एंटीबॉडी थे से जाके कोट वाला एंटीजन सहित बाइंड करेंगे तो ताको सेकेंडरी एंटीबॉडी बाइंडिंग भी बहुत हम आ से हिसाब रे कलर प्रोडक्ट भी बहुत हम सो मोर कलर प्रोडक्ट मीन लेस antigen in the original sample. So there is a inverse relation between the color product and the antigen concentration in the original sample. Is it clear now?
Yes, ma'am.
Yes,
Okay,
ma'am.
you can go through the text once again. If you still find it difficult, then I guess it will be clear. So Ria, जो radio immune आसे, it is also similar. Maybe एटा देखिले maybe टिके confusion clear ही परे. This is also similar to sandwich ELISA. देखो. This is called RIA or radio immunoassay. Okay. So here also what you are using the radioactive antigens. Okay. So this is the first antibody that is tagged with your radioactive antigen. Now this is the on-labeled antigen. Now if the concentration of this on-label antigen will be more, ताले कौन होगा? They will go and they will replace the radio-label antigens. Okay. Then you need to precipitate the antigen antibody complexes with the secondary antibody set the precipitate column. Then you measure the radioactivity of the bound antigen. So the higher will be the radioactivity of the higher will be the radioactivity of the supernatant. That means the more is the the lower is the concentration of the radio label antibody. See, okay, some people have raised your hand. You have any question, or it's by mistake only? Mona Lisa Rautray. So, किसी question अच्छी कि एमती ही by mistake hand raise करछो? See, देखो ये कौन होची? It is similar to your competitive ELISA. देखो ये जगह रे तमे already कौन use करछो? You are using a label radio label antigen. ना? So antigen को already antibody से तो bind करी के तमे use करछो. Then you are adding the on label antigen. Suppose तमे patient रू जोटा isolate करिछो, you do not know कि तादरे antigen अच्छी कि नहीं तमे जानी ना, but you are suspecting कि जो रेडियो लेवल एंटीबॉडी तमे यूज करिछ एठी से पेशेंट रो सैंपल ले से ऑन माने सस्पेक्टेड एंटीजन अछि ताहले एबे कोन हबो ना ऑब्वियसली जोटा हमर माने ऑन लेवल एंटीजन अछि तार बाइंडिंग एफिनिटी इज मोर इन कंपैरिजन टू दिस रेडियो लेवल बाइंडिंग एंटीजन सो जदी ए जो ऑन लेवल एंटीजन रो कंसंट्रेशन बेसी हबो से कोन करिवो सी ए जो रेडियो लेवल बाइंड एंटीजन अछि ताको रिप्लेस करबा को चेष्टा करिवो एते जाय बुझ परछो कि नहीं
Yes, ma'am.
so there will be also a kind of competition hela so jo on level antigen achi it will try to replace the radio level antigen that is bound to the antibody tapre e jo bound antibody ko tame precipitate karo using a secondary antibody acha एबे कौन हबो ना जदी तमरो ऑन लेवल एंटीजन रो कंसंट्रेशन बेसी थिबो ताले से माने कोन करिवे बेसी रेडियो एक्टिव एलिमेंट को रिप्लेस करिथिबे एथरे जे कि सेकेंडरी एंटीबॉडी रे बाउंड हिची ता माने तमरे जो सुपरनेटेंट कहले कोन ना प्रेसिपिटेट करबा माने सपोज गोटे सॉल्यूशन अछि ताको सेंट्रीफ्यूज कर दियो प्रेसिपिटेट माने जोटा बसी गला सेटा होची तमरो प्रेसिपिटेट हला आ जोटा ऊपर बाहर पानी बा लिक्विड सॉल्यूशन जोटा रहि गला सेटा होची सुपरनेटेंट सो ताहले जदी तमरो ए जो ऑन लेवल बा ब्लू कलर एंटीजन रो कंसंट्रेशन बेसी अछि माने सेगुडा बेसी कोन हेबे बेसी बाइंड करि जिवे एंटीबॉडी को ताले तुमको सुपरनेटेंट रे कोन बेसी मिलिबो सुपरनेटेंट रे बेसी तुमको रेडियो एक्टिव एंटीजन गुडा फ्री मिलिबे हला एति की बुझ परलो कि नहीं फ्री रेड गुडा फ्री बेसी काहे के हेबे जदी तमरो ब्लू डाक रो कंसंट्रेशन बेसी हबो ताले कोन से माने तो सब एंटीबॉडी रे बाइंड करि जिवे ना बाय रिप्लेसिंग द रेड एंटीजेन्स और द रेडियो लेवल एंटीजन व्हिच वाज ओरिजिनली बाउंड टू योर एंटीबॉडी तो एबे तमरो सुपरनेटेंट को तमे जेतले रेडियो एक्टिविटी पई चेक करिवो यार रेडियो एक्टिविटी विल बी हायर so means more the radioactivity of the supernatant higher is the concentration of your suspected antibody माने blue color antibody जोटा labeling हेइनी सिमिलरली जदि तमरो पेशेंट रे लोड बहुत कम अछि ताले कोन से ब्लू गुडा जाय के रेड को आउ रिप्लेस करि परिबेनी ताले तमरो सुपरनेटेंट रे कोन थिबो रेड रो माने रेड एंटीजन रो कंसंट्रेशन बहुत कम थिबो सो रेडियो एक्टिविटी विल बी लेस ला so less radioactivity means what we can say the less radioactivity means this sample concentration or the unknown sample concentration will be less hela so that is how you need to find out the concentration of antigen in case of radioimmunoassay so jehetu etu tome radioactive element use karucha you are calling it as radioimmunoassay. It is similar to your competitive ELISA actually. So you can read the text once to clear if any further doubt is there. It is difficult, right? It is a little bit confusing. जेहेतु first time hele टिके confusing लगी पारे, but तुम पढ़िले देखियो you can easily understand this.
Ma'am, the competition binding is occur due to affinity binding.
Yes, due to affinity of the antigen towards the antibody. Usually, amaro ei jo antigen achi na, yar affinity will be higher than this radio label one. So they will try to replace them. Yes, kono question?
Radio level antigen is bound with wall of
sorry तुम voice से clear सुना जाऊनी
radio level antigen is bound with microrotor plate
no here it is bound to the antibody actually तमे microtiter plate रे दबा पूर्वरु already antigen antibody रो mixture तैयारी कर दिछो you have mixed the radio level antigen or radioactive antigen with the specific antibody तापरे से antigen antibody रो mixture को microtiter plate रे देलो आ तापरे you are giving the suspected antigen ला जोठी तमे allow करछो के they should compete and replace each other or they should bind माने compete for binding with the respective antibodies okay see I have put some of the questions but let me move to that now next we have this characterization of the clone by sequencing method so I think I will skip this part because this is not actually very much related to this but I hope tamane Sanger sequencing sunicho ki nahi Sanger method chain termination method sequencing by chain termination method so just mu ଟିକେ माने येटा टेक्निक एक्सप्लेन कर दोची बट यार एते भी किछ रेलेवेंस एठी नाही सी हमर कोन करा हुए ना जेतले तमे एटा एक्चुअली बहुत अर्लियर माने सीक्वेंसिंग माने कोन हमें जो डिकोड करूचे जो डीएनए रो जो बेस सीक्वेंस ता कोन अछि माने एटीसीजी जो अछि सेटा केमती सीक्वेंस अछि वी आर ट्राइंग टू फाइंड आउट दैट इज कॉल्ड एज योर सीक्वेंसिंग so sequencing by हमें कौन यूज करा हुए तमे गोटे पर्टिकुलर जीन ऑफ योर इंटरेस्ट नेला ता संगे गोटे प्राइमर माने which is having some basis complementary to the template DNA or your target DNA तापरे what you are using in the solution is instead of DNTP you are using DNTP I hope समस्ते जानी थिबो डिऑक्सी न्यूक्लियोटाइड ट्राइपेप्टाइड जो गुड़ा अच्छे डीएनटीपी माने डीएटीपी डीजीटीपी जीसीटीपी जोड़ा कि हमरो गोटे गोटे तमरो ऐड करि चलिबो कॉम्प्लीमेंटरी बेस पेयरिंग रे नुवा डीएनए सीक्वेंस पे हला सो से जगह रे तम आमे डीएनटीपी सहित कोन यूज करछ कि यू आर यूजिंग डीडीएनटीपी सो डीडीएनटीपी कहले कोन ना डीएनटीपी जोटा अच्छे सेटा तमरो सीक्वेंस को कंटिन्यू करिवो किंतु DDNTP हेले कौन हबो ना इट विल ब्लॉक द चेन माने इट विल ब्लॉक द कंटिन्यूएशन ऑफ द चेन से कि मेथड रो ना कोन दिए छि चेन टर्मिनेशन मेथड हला सो सपोज एठी अछि A T C G ताले कॉम्प्लीमेंटरी बाइंडिंग कौन हेबा कथा इटी कौन हेबा कथा T तापरे A G C लाइक दिस हवा कथा तो कौन हुए ना एटा जदि तुम DNTP लागिबो ताहले तुमरो चेन टाइम इलोंगेशन हे चलिबो या पर सपोज A अछि कि C अछि अकॉर्डिंगली तुम चेन टा इलोंगेशन हे चलिबो बट जदि तुमरो ए DNTP जगह रे DDNTP लागि गला ताहले चेन टा कोन हिजबो ब्लॉक हिजबो सो कोन हुए अब ई जगह रे ना सपोज इंस्टेड ऑफ माने हमर अछि सीक्वेंस A C G T सपोज अच्छी एमती ताले तुमरो एट जगह कौन लगिला सपोज T आसिक लगिला तापरे जदि एठी C जगह DDGTP लगिगला डाई डिऑक्सी गुआनोसिन सो ये कौन करिवो ना आगो को बढ़िबा को दबनी तो तुमरो ए जो टेम्पलेट डीएनए अच्छी ए टेम्पलेट डीएनए रे कौन हबो ना जहाँ भी थिबो ATCG जेमती सेही हिसाब रे गोटे गोटे सेगमेंट तयारी हे चलिबो T खाली आ गोटे कौन ना गोटे सेगमेंट खाली हे परिबो TG आ गोटे सेगमेंट कौन हे परिबो ना TGC लाइक दिस हैला तापरे ताकू कौन करा हुए ना हमर जेमती आमे देखिला जेल इलेक्ट्रोफोरेसिस रे से सेगमेंट गुडा को आमे एमती साइज वाइज ताकू माने साइज वाइज ताकू एमती तमे अरेंज करि परिबो हा तापरे अरेंज करि पाले कोन ना तमे गोटे पर गोटे बेस पेयर एमती जोड़ी दे गले तम कोन मिलिबो यू कैन गेट द सीक्वेंस ऑफ द डीएनए हैला एटा क्रोमेटोग्राम ना दिया छि काहे कि ना तमे देखि पारबो ईच ऑफ द DDNTP इज लेबल्ड विथ अ डिफरेंट कलर टैग कोडा रेड कोडा ग्रीन कोडा ब्लैक कोडा ब्लू एक्चुअली यार भी बोधे फिक्सेशन थाए माने टी टा रेड ए टा ग्रीन एमती किछि कलर कोड भी थाए तमे जेतले सीक्वेंसिंग करा हुए सेमती ही कलर कोड यूज करा हुए तो जो हिसाब सपोज एठी टी अछि तो टी रे स्टॉप हे गला तापरे एजी जदि DDNTP लागि गला तापरे आगो को प्रोसीड ही करिवनी सो एमती तमरो डिफरेंट सेगमेंट्स ऑफ डीएनए तयारी हिजबो आ अल्टीमेटली तम तक जेतले जोड़ीबो यू विल गेट द सीक्वेंस ऑफ योर टेम्पलेट डीएनए हला जेतु एडी चेन टा टर्मिनेट हे कि तमरो सीक्वेंस टा में पाउचे दिस मेथड इज कॉल्ड एज चेन टर्मिनेशन मेथड
Ma'am, now this method is used to sequencing.
it is used used for sequencing but actually हमरो आउरी भी बहुत एडवांस टेक्निक गुडा आसी गलानी जेमति हमरो सीक्वेंसर भी आसी गलानी बट प्रिंसिपल रिमेन्स ऑलमोस्ट द सेम बट नेक्स्ट जेन सीक्वेंसिंग हमरो आसी गलानी जोथरे हमरो माने तमे एट ए टाइम बहुत गुडा डीएनए रो सीक्वेंस गुडा बाहर कर दे पाछो but still eta ebe भी use रे अछि but this is one of the first techniques developed for sequencing of DNA इवन एटा अमीनो एसिड रे भी सेम माने प्रिंसिपल यूज करा हुए टर्मिनेशन मेथड तापरे अच्छा हां someone was asking कि केमती हमरा इंसुलिन तयारी करा होची ओके ह्यूमन इंसुलिन केमती तयारी करा होची सो इटी देखि परिबा actually insulin is a hormone that is produced by the beta cells of pancreas la pancreatic beta cells so bacteria re actually insulin the gene to nothibo so what we have to do we have to transfer the human insulin gene into the bacterial cell so that and moreover jo tumko vector bisare kahithli so vector are actually of two different types of vector could be there one is called as the cloning vector the other one is called as a expression vector la so cloning vector kam kona na ye kebala simply it will carry the segment of DNA and it will simply help in replicating that DNA segment but expression vector what it can do it can actually lead to expression of the gene product na so ei jaga re amar kintu expression vector darkar because we need the protein or the polypeptide from the gene of our interest so it is dekho technique kemti kora hochi what are the steps so first you are means isolating the vector so this is the vector which is carrying the promoter the insulin gene A subunit then there is another vector has been constructed that carries the insulin B subunit So actually dita polypeptide chain thai insulin ra. So one vector is carrying the A subunit, the other vector is carrying the B subunit. This is the antibiotic resistant, that is the marker gene for selecting the transformed bacteria. And this is the lac gene that will actually carry the transcription of this insulin gene along with the lac operon. So the beta gal or insulin A fusion protein accumulates in the cell. ଯେତେବେଳେ ତମର lac operon express ହଉଛି alongside other lac gene, the insulin gene A will be also produced. Similarly, in the other bacteria, insulin B gene will be produced. Now, you can simply extract the protein by separating it from the rest of the bacterial component. So you need to extract and purify the beta gal or insulin fused proteins. Then you can treat them with the cyanogen bromide to cleave the A and B chains. So cleave means actually amara gote cyanogen bromide hochi one type of the mane kon kaya protease mane je ki protein ko cleave kari pare. So ये जो pro insulin part अच्छी actually that pro insulin part needs to be cleaved so that the A and B subunit can combine by the disulfide bond to form the active insulin. So that is how the insulin is being prepared by using the transgenic method. जोथरे हमें कौन करछे जीन दीटा को जो पॉलीपेप्टाइड अछि जोटा दीटा अलगा अलगा जीन रो हमरो एनकोड होछि. We are transferring the genes into two different vectors. Separately, we are cultivating the A chain and the B chain of the A subunit and the B subunit of the insulin polypeptide chain. So, A jo jaga dekhcha, this is actually called as the pro-insulin. So, in the pro-insulin, this polypeptide needs to be cleaved or it needs to be removed for formation of the active peptide. So active A or B peptide form hela pare ta bhitare taku fuse kariki gote disulfide bond ta bhitare form hoba, then that is converted into the active insulin. Then that can be taken or used by the humans directly. So jeta simple dekha, it may not be that simple. A lot of experimental processes are involved in that.
Ma'am, as the human protein is produced inside the bacterial cell, but
Yes.
What about the protein folding? Because
The
these
protein
are
folding. See, the bacteria is also having its own protein folding mechanism, right? The protein actually, the primary sequence, whatever is there, the primary sequence itself holds the all the information for the protein folding. Okay. So since the bacteria is also having the same cytoplasmic constituent, so there is no problem in folding of the protein. It will be folded properly. Means all the protein may not be folded, but for insulin it works fine. For protein folding, actually not much external things are needed. The protein can undergo its own conformational change to form the active form. Okay.
ma'am,
So
the
it is
bacteria will not integrate into the bacterial DNA.
Sorry, the bacteria.
The vector we inserted our desired trait.
No. Why it will not integrate? Because this is we are targeting it to a plasmid DNA. Not all the plasmid DNA of one bacteria can integrate into the genomic DNA. Okay, some plasmids can integrate, but not all the plasmid. So whatever plasmid vector we are using, it does not have the a tendency to integrate into the bacterial genome. Then this is the construction of a transgenic plant. I hope all of you might have heard about this Agrobacterium tumefaciens that has a very unique kind of very unique kind of DNA called the tDNA. So this is the step involved in the construction of a transgenic plant. So transgenic plant or transgenic animal simply means the plant or animal that carries a gene from carries a foreign gene from another organism. Okay. So here you can see this is the microscopic view of the bacterial cell. This is the bacterial chromosome. This is the plasmid. So this tDNA, it has to undergo the restriction cleavage where it can actually carry the foreign DNA. Okay, this is the foreign DNA caught with the same enzyme. The tDNA has to be caught with the same restriction enzyme. So this is important to remember, because if the enzyme will not be same, then the caught produced will not be same and the ligation will not be happening. Okay. Then you can insert the gene into the plasmid using the restriction enzyme and the DNA ligase. Then there is formation of this TI plasmid, that is recombinant TI plasmid, which is carrying actually the gene of your interest, this blue color. This is the promoter gene for expression. So again, we can say this is a expression vector, not only a cloning vector. Okay, because it will actually express the gene of your interest into protein. Then you can introduce it into the plant cell. Then you can culture the plant part and you can form the plantlet. And when you grow the plant will already carry the characteristic of this particular foreign gene. Okay. Like let's say there is one example I think I have cited here, that BT cotton example, to make the plant resistance against the Bacillus thuringiensis, the pathogen is there. So this BT resistant, this is the cotton plant that is resistant to the insect cells. So these are the cotton cells. It will produce a prototoxin. The toxin is cleaved and is converted into a toxin, where this toxin can create pores in the gut. Gut means the intestine or the digestive tract of the insect, and the insect will die. So that is how the plant will attain what we can say resistance against the pathogen. Okay. So
Ma'am, the
here we can
plasmid will not get destroyed inside the cytoplasm
where in the
in plant
bacteria.
cell. No, in the plant cell.
Wait, no, it will not usually be destroyed, because okay, so here, no, it will not be destroyed. See, this is actually the plasmid is a extra chromosomal DNA. Okay, so the plant need to have some reason for killing this, and this TI plasmid, actually, why this TI plasmid is used? This is actually a infectious pathogen for the plant. Okay, so means see, there is a host pathogen relationship. So since this means a plasmid can already infect the plant, but here instead of carrying the virulence gene, it is carrying a useful gene. So that is why they are compatible. It's not like the plasmid will be inserted and it will be destroyed by the plant. No, the plant is already compatible with this plasmid vector. Okay, so it will
Ma'am,
not.
there might be nucleus inside the cell, so that can be damage the nucleic acid.
No, see, the nucleases are there in the cell, but the cell, the means the plasmid or the means the nucleic acid, they also have certain techniques to protect themselves. Like we will say in case of, we will see in case of CRISPR. Okay, so that is that I will discuss ahead. So although see, if that would have been the reason, then no nucleic acid would be surviving inside any cell, because every cell will be containing nucleate. So as there are certain means as the nuclease has the capacity to degrade the nucleic acid, similarly the nucleic acid also has certain type of means rescue technique to protect itself. Okay, so it's not like randomly every nuclease will cut means every nucleic acid. So there are certain means markers are there, so that is how they will be protecting themselves. So
Ma'am,
like we saw,
every cell will allow the plasmid. Every cell membrane will allow the plasmid.
no, it may not be possible. So that we have to find out experimentally. It may be possible, it may not be possible. See, I told you this is allowed because this is actually a natural pathogen for the plant. The TI plasmid, it is a pathogen for the plant. So they are already compatible. Okay. So like for example, if something is injected into the mosquito and is allowed to insert into human, then we won't have any problem because we are already compatible. That is already a parasite to us. So it's something like that. Okay, so they are already compatible. That is why that plasmid is being used. Okay, then coming to this transgenic means animal, like we saw the transgenic plant. Similarly, the transgenic animals can be produced. So this is a very common example of the formation of the means the seep that was first formed by the nuclear transfer. Okay, so the same thing is being explained here. I don't think it is of much importance. Here is one example that you need to see, that is the development of a enviro pig. Okay, so this pig is actually very much environmental friendly. Why? Because this has been transgenically designed or genetically modified to reduce the phosphate accumulation. Okay, so here you can see these are the food that is the pig is consuming, but inside its digestive tract, and as you can see, there is a phytase P. Okay. So this enzyme or the gene containing this enzyme, encoding this enzyme, has been introduced into the pig. So what this will do? Now this can very efficiently catalyze, or it can very efficiently metabolize the phosphate formation. So when the pig will be releasing the excreta will have extremely low amount of phosphate. So that is how it is useful in reducing pollution or eliminating phosphate contamination from the soil. Okay. So that is why this is what is called as a enviro pig. This is like environmental friendly. Similarly, many animals and plants can be modified as per the requirement. Okay, so they are called as transgenic plants or animals. So many examples are there, like we are using the technique to enhance the production of the plants, or even enhance the quality content of milk, or enhance the fat content in some kind of animals, protein content in animals. Like that, we are using the genetic engineering to create transgenic plants and animals. Okay. The next
Ma'am, is
coming
there
to
any side effect by consuming the GMO food product?
actually, that could be a controversial question. So it should not be in means until and unless it is affecting means the physiology of human beings, there is no harm in consuming those kind of plants or animals. Okay, so but if it depends upon exactly how you are designing the plant or animal. So if it is only for fortification, like adding something more or increasing the productivity, it should not be a problem. But if like somehow whatever means things you are adding to the GMO, if the humans are unable to digest it or something like that, or if it goes on and interferes with the normal metabolic activity, then there could be a problem. But until then, it should be fine only. Okay. Rather, it is more useful actually, because looking at the supply chain demand in current scenario, GMOs are more preferred. But the thing is, they have to pass through whatever means what we can say safety protocols we have. They must be certified through that. Okay, so until and unless the GMO is actually certified, means it is a certified product, that could create some kind of problem. But if it is following the proper protocol, and if it means gets passed through the certification, then there should not be any problem, because they are already under the permissible limits. Okay.
Ma'am, is there any possible the GMO gene will inserted into the human cell?
No, no. See, when you are eating something, no, you are not taking the DNA. You are taking it in like food form. So there is no possibility how it will integrate into your genome. You are not taking the vector or anything directly. So there is means almost nil chance. It's not possible. Okay, let's proceed. The next, very one of the most interesting concept that is developing these days is the CRISPR. So I hope everyone is familiar with the term CRISPR for gene editing. So even like recently, the Nobel Prize has been awarded for the for means elucidating the mechanism of CRISPR or Cas9 activity. So CRISPR simply stands for clustered regularly interspaced short palindromic repeats along with CRISPR associated or Cas gene that impact the adaptive immunity in some bacteria and archaea. They protect the bacterial genome from exogenous invading DNA elements such as bacteriophage, foreign plasmid DNA, etc. It was reported that the integration of a DNA fragment of bacteriophage at CRISPR locus of Streptococcus thermophilus made it resistant to a bacteriophage. So the CRISPR region contains short palindromic repetitive base sequences. They are separated by variable sequences called as the spacers. The spacers have the sequence homology with the DNA of the bacteriophage, plasmid, or many other exogenous DNA. The Cas genes usually flank CRISPR array that is preceded by the leader sequence. The Cas has exonuclease activity. There are three types of CRISPR, of which the type 2 is the most detail studied. Okay, so exactly what happens in CRISPR, let me explain you. See, actually, maybe I think that diagram is not like I have forgot to, means it has been missed by me. The CRISPR means the how exactly the CRISPR looks like. See, the CRISPR is actually made up of short DNA sequences like this. Okay, and that to palindromic DNA sequences. So what happens when and there is another enzyme that we discussed that is what Cas CRISPR assisted enzyme or CRISPR associated enzyme Cas9 is there. So what it does when any foreign DNA enters into the bacterial cell. So what the Cas9 does, it simply degrades that, cleaves that particular DNA and degrades it so that they can no longer integrate into the genome of the bacteria. Now, as you were asking, so how this Cas9 is not acting on the bacteria itself? Because they have certain methylation points. So the Cas9 can easily discriminate which is the host DNA and which is the outsider DNA. So if in the phase DNA there won't be any methylation patterns, so the Cas9 can easily understand that this is the means foreign DNA which I need to cleave. So some part of this means CRISPR genome has homology with that of the phage genome or viral genome. So since the Cas9 can means identify this CRISPR sequence, it becomes easy for the Cas9 to identify the foreign sequence as well. So the only difference how it will discriminate the host DNA and the means DNA of the outsider is through this methylation pattern that are actually marker to protect its own DNA. Okay. So how the CRISPR is used for targeting means gene therapy or how it can be used for repair of certain kind of gene. So here a small technique is being means described. So a short guide RNA is essential for means targeting the Cas9 or taking the Cas9 to the site where the gene insertion is happening. Okay. So there is another sequence called PALM. Okay, so the Cas9 will it will be actually guarded by or guided by the guide RNA and will brought to the site where you need to carry out the gene replacement or you need to insert a gene of your interest. So at that site the cleavage is being done and after the gene is cleaved you can simply insert the donor DNA and as these two sites will be homologous to each other there will be homology directed repair of the DNA and now this DNA is having, or this segment is having, the gene of your interest. So this can be the future of gene therapy, where if there is any kind of defective gene is present, that can be easily cut, and the defective gene can be replaced by your means rectified gene sequence or the normal gene sequence. So like for example, as we are having some inborn kind of a problem, like let's say sickle cell anemia or hemophilia, where there is means minor genetic mutation is there, that is creating the problem or that is forming the defective gene. Those genes can be rectified by this CRISPR technique. Okay. Similarly, even if there is no homology is there, the insertion or deletion might be directed by non-homologous end joining. So these are actually some of the DNA repair techniques which you can refer and read in detail. So it's not possible to explain over here. So just remember, these are the techniques that are adopted by the DNA to complete the ligation after the insertion of the rectified gene sequence or after the insertion of the foreign gene sequence. Okay, so this could be also a question for you, the CRISPR or the mechanism of CRISPR or Cas9 activity. Okay, it may not be a long question, but it could be a 5-mark question for you. Okay. Then coming to some of the last parts, that is your tissue engineering and gene therapy. So tissue engineering is simply utilizing the technique for improving the biological system. Okay. Okay, so first I think tissue engineering is left. So gene therapy. So what is actually gene therapy? So the interchanging a disease causing gene with a healthy copy of the gene, inserting a healthy gene to replace a non-functional or dysfunctional gene, or repairing of an abnormal mutated gene by selective reverse mutation. So these are the ways by which the gene therapy is being carried out. So as you can say, it is carried out by two ways. One is called as the viral method, another is the non-viral method. So viral method means where you will be using a virus as a vector to carry your gene. Like in case of humans, we mostly the adenovirus is used. And for non-viral method, methods such as a gene gone method can be used, where directly the DNA will be inserted into the cell. Okay. So the vector that contains the normal or the healthy gene. So since, as I told you, this vector will be a pathogenic vector, or it is actually a vector that can infect the human cell. So there will be compatibility, and the genome of this viral vector will have some kind of homology with the genome of the with the target genome. Okay. So there will be like integration of the viral genome or integration of this new healthy gene into the genome of the target organism, where the means the non-functional gene will be replaced by the functional gene, and the disease can be cured. So this is an example of in vivo and ex vivo gene therapy, how it is carried out. So in vivo means inside the laboratory, and ex vivo means in vivo means not exactly in the laboratory, means outside the body, and ex vivo means inside the body. Okay. So in ex vivo, what you can see, there is isolation of the defective cells. The cells are grown in the culture. The vector containing the therapeutic gene is means it is subjected, or it is integrated or infected with the cell that you are growing. The culture cells are transduced with the vector expressing the therapeutic gene. The therapeutic gene containing the cells or the genetically modified cells are reintroduced into the human or into the target organism. So this is how ex vivo therapy works. So ex vivo means in the laboratory. Okay. Then in vivo means you have the therapeutic gene, you are inserting the therapeutic gene into the vector. You are infecting the human with the vector directly. And since the vector genome will have some kind of homology with the genome of the human, then it can directly go and replace the defective gene here directly. So that is what is in vivo method. In vivo means you are using the organism directly. So in either way, the defective gene can be rectified. So how exactly the gene therapy can work? So mostly it can be used for cancer, or also it can be used for in dermatological cases. The skin is the largest, more superficial and easily accessible organ of the body, due to which its observation and manipulation is easy. It serves as one of the most desirable organ for gene therapy. So recently, several attempts have been made to treat the skin diseases by gene therapy, such as your epidermolysis bullosa. Then there are certain type of melanoma, etc., that can be treated with gene therapy. So since it is a superficial organ, it is easier to target the organ and carry out the in vivo kind of experiment, or even ex vivo kind of experiment. Similarly, for cancer also, the gene therapy can be used. Like if you identify the cancerous cell, and if you can know exactly which gene has been mutated and that is leading to cancer, that can be rectified through the insertion of the new gene, or to the normal gene. Then we come to the end, that is the tissue engineering. So tissue engineering, like use of techniques. So when you are using the engineering means a technique for generating some healthy cells, or generating or designing some cells of your own interest, that process is called as your tissue engineering. So what actually happens in tissue engineering? You can isolate some of the cells, you can culture and you can differentiate them, then simply you can grow them in a scaffold. Scaffold means that is actually a supportive material. Okay, so it could be natural or it could be artificial also. Then you can use this as a graft and you can implant it to the a person whom you want to treat, like particularly for burn injuries. Okay, or means replacing some of the organ that has been destroyed, the tissue engineering can be used. But there are a lot of ethical issues are associated with the tissue engineering, like how to use this, where to use it, how far it is means affordable to all, or if there is any kind of misutilization of the thing. So a lot of means if you are working in the field of tissue engineering, you have to you have to go through a lot of ethical practices. So this is one of the example of tissue engineering, like in regenerative medicine. They help in repairing the damaged tissue or organs of the body by stimulating the body's self-repair mechanism. If the body cannot heal by itself due to physiopathological condition, tissues or organs can be grown in vitro, followed by their implantation in the patient's body. So regenerative medicines also include the use of stem cells or progenitor cells for the formation of specific cell type through directed differentiation in a highly controlled environment. Some examples are like tissue engineered skin is being used to improve the wound healing in case of severely burned patients having ulcers or chronic wounds and acute injuries, etc. In recent years, the tissue engineering has emerged as one of the better options to restore the function of the damaged renal and cardiac tissues, etc., through the implantation of tissue engineered grafts. So you can also use the stem cells, like the undifferentiated cells. So you can supply them with your adequate growth factor that will lead to development of certain type of cells, if you need to have this, let's say kidney cells or liver cells, something like that, you can direct their growth, and those cells can be directly implanted to have a repaired or regenerated organ. So these are some of the technological aspects for the welfare of the human beings. But both your gene therapy and tissue engineering, they have a lot of ethical complications. Okay, so you must pass through certain ethical tests in order to carry out the experiments. So is there any question from you till now, or we can discuss few of the questions that could be important from your exam point of view, how you should be like writing the questions, and then we can wind up. If you
Ma'am, can
already,
you give the permission? One question I asking today class not related this question.
sorry, I like your voice is not very clear.
Ma'am, it's one question not related to today's class. Can I you give me permission? Then can I
Yes,
ask?
yes. Now see, since the class is over, you can ask whatever you want. See, why I am stopping you in the middle of the class is maybe that question is not related to others, and they might think that their time is being their time is being wasted. So at the end of the class, whoever is having any query, you can stay back and you can discuss with me. And those people who
Okay.
those people who do not have any question, they can leave. If they want, they can leave. Okay. So there is no problem in you asking the question or anything. You are free to ask whatever you want. But
If
please correct your background. It's creating a lot of noise. Okay, means the question is not at all audible. So please, mm, the voice is not very clear. So please, silent the background and then ask your question. Or you can simply write also. I will read and answer that also. You can write also if you want. Okay. Okay. So these are few questions which are which are mentioned in your assignment. I don't know if you are getting the same assignment or not. ये same assignment questions तुमको इथर मिली छे. इटा last year मु pick up करथली. So this time also you have the
Yes, yes,
same
ma'am.
assignment.
Aita. Yes, ma'am.
Same assignment. Okay. So also you can see there are means a few sample questions are there which might be important for your government examination. Okay. So the first question, I don't think it is much probable, but a few questions I would also like to mention. Like this is one of the very probable question, the linkage and crossing over. Okay. Then this one, mutation. So what is mutation? And what are the general classes of gene mutation? So if it is about gene mutation, you don't have to write about the chromosomal thing. Okay. But similarly, you can also have a question like explain different type of chromosomal mutations. So chromosomal mutation means if it is only about chromosomal aberration, you have to mention both structure and number also. So structural, like you have four different types, that deletion, duplication, etc. You have to mention, and the numerical you can write about that aneuploidy, polyploidy, etc. Sorry, that aneuploidy, the trisomy, monosomy case, whatever is that, you have to explain that part. Okay. And you can. This question was already asked in the last exam, I guess. So it may not be that important. Then this one could be an important question. Okay, that development of transgenic plant or even the insulin one. Someone was asking regarding the insulin. So the insulin question could also be important for this time. Okay, Parvati, did you write your question or you want to ask? Admission after MSc. No, MPhil. See, these days MPhil is means it has been cancelled only. The degree is no longer valid. Means the MPhil has been completely eliminated. Okay, so after MSc only you are eligible for PhD. But one thing is important, that is you must have any of the you must clear one of the fellowship exams like either CSR, NET or GATE. Although in some of the universities you can take admission through their examination process, but I don't think these days anyone prefers to take a student who does not have a fellowship. So it is always advisable to clear one of the fellowship exams before you get admitted into a PhD. Okay.
Ma'am,
MPhil has nothing
ma'am, I
with
want to
PhD.
ask something.
Yes, you can
Good
ask.
evening, ma'am. Ma'am, since the question will carrying 10 marks, how much pages we should write?
See, okay, I have some sample answers I want to show you, okay, from your last year. Okay, see, there is one question I told you. This one, question number 8. Look at question number 8. The question is write a note on diseases and syndromes due to changes in the chromosome numbers. Okay. So, to answer this question, first you have to explain what is the normal chromosome condition. Like in humans, what is the normal chromosome condition? So, you first write 2 lines regarding that, that we have this 23 pair of chromosome, 22 pair autosome, and all those things. You write 2 to 3 lines. Okay. Then you must explain this. How exactly the problem in the number is arising. So either you explain it in your words, but my suggestion is instead of explaining, it's better if you are writing like this. Sorry, if you are drawing a figure like this, because it clearly demonstrates how the things are happening and what is happening. Okay. Then next, what you have to do, you have to cite the examples. So first you cite some autosomal examples. So since the question is for 10 marks, you must include at least 5 to 6 type of disorders. Okay. So autosomal, you have 3 major types. You can mention that, like your Patau syndrome, Edwards syndrome, and Down syndrome. Similarly, in the sex chromosome aberration, also you have 2 types: Klinefelter and your what we can say Turner syndrome. So all the 5 types you mentioned, like in which kind of chromosome means which type of numerical aberration is there because of which this is happening, and you must mention 4 to 5 symptoms. What will be the possible outcome if this abnormality is there in the person? Okay, so that will be like enough for a 10-mark question. You explain what is the normal condition, then you from normal you go to the abnormal, how exactly the abnormal condition is developing, due to what mechanism. Then you cite the examples where you are observing the anomaly, the 3 autosomal anomaly, trisomy, and 2 sex linked or allosomal anomaly you explain, along with the like which chromosome is responsible, in which type of individual it might occur, what is the frequency, like what is the possible chances. Okay, and then you explain few symptoms. So that is enough for the 10-mark question. Similarly, I have 2 or 3 more examples. Let me show you. So these are some of the questions from last year. So what are the steps of gene cloning? So and when you are answering the question, just look at the fraction. So when the means the marks are awarded in fraction, you must take care to answer the questions in fractions also. Okay, so if you elaborate one of the fraction and you skip the other one, you lose that part of the mark. So first you answer what is gene cloning. Means 2 to 3 line you write down what is mean by gene cloning. Then you write the steps. There are 7 steps of gene cloning, like isolation of the target DNA. Then first is restriction digestion of the target DNA. Then isolation of the DNA. Then restriction digestion of the vector. Then ligation of the target DNA into the vector. Then multiplication of the transfection to the host genome or the host cell, that is the bacteria or E. coli, then multiplication of the bacteria or E. coli. So these are the steps of the gene cloning. Then describe the different DNA manipulation enzymes. So there are a lot of manipulation enzymes are there that you must mention, like 2 to 3 lines you must write about each nuclease, what is the function, in which type of DNA they will be acting, what is, like how exactly what is the mechanism of action. Then what is S1 nuclease, Mung-Bean nuclease, ribonuclease A, ribonuclease H, DNA ligase, DNA polymerase. Okay, so these are the DNA manipulating enzyme, including some of the DNA modifying enzyme like your alkaline phosphatase, T4 polynucleotide kinase, terminal transferase. So you must you don't have to write all these things, but means from this summarize 2 to 3 lines, that is actually reflecting the function of the enzyme, and you mention that. Okay. Similarly, there is another question that is like the production of the growth hormone, human growth hormone. Okay. So this is the step of production of the human growth hormone through this transgenic means technology, genetically modified through genetically modified bacteria. So first you must write something in textual form, like why this growth hormone production is essential, why you need to do it through transgenic, through genetically modified organism. You first mention that. Okay. Then 2 to 3 lines regarding the importance of growth hormone in humans. Then you write down the steps. Either you write it like in this arrow manner, you can write it down and you can draw the diagram, that will be enough. Or if you need to write in textual form also, you can describe. But always it is preferable that you draw a diagram like this, because this clearly illustrates the entire procedure. Okay? And in a few things you write in the textual form. So these are the few things I wanted to inform you. I think I already mentioned what could be the possible important questions. From this part only you can choose. Like I don't think you will be getting questions from this, but you might get question like lethal alleles and epistasis. These you can expect as small short questions. Okay, like 2.5 mark or 5 mark question you can expect. Then for long question, this could be a possible long question for this one. The fourth one, then fifth one, then eighth one, okay, 8, 9, and 10 also. Since you did not have any question on CRISPR or Cas9 targeted genome editing, you might expect this also. So these could be the few important questions. And apart from that, I think mutation part will be important. Then what else is there? Tissue engineering could also be a question for you, or gene therapy or tissue engineering. Like that, you have to prepare. Okay. And I think I have already shared the PPTs for the rest of the 3 classes. This one I will share now. So, is there any other question from anyone? Yes.
Diagrams. How many pages should I write in 10 mark questions?
Sorry. Can you repeat? How much?
And how many pages should I write in 10 mark questions?
See, the thing is, in science, the page, you should not think about page. You should think that how correctly you are answering the question. Okay. So it's not like that if you are writing many pages, you will be getting good marks. It's not like that. Whatever question is being asked, your answer should be precisely mentioned in the in your means presentation. That is what is matters. Like here, as I told you, the example that I cited here, the question here is, look at the question. The question is what? Explain the methodology of production of the recombinant growth hormone with the help of a suitable diagram. It is already asking for a diagram. Okay. So if you only draw this part, then also that is enough. The examiner will be very happy to award you the full mark, because it clearly demonstrates all the steps, all the procedure that is involved in the product, in the development of the human growth hormone. Okay. So you have to design your question in such a way that whatever question is being asked, your answer is all the content are there in your answer. Okay. So that is how you need to design. For 10 mark, I think 2 to 3 pages should be enough, including your diagram.
Ma'am, we have to draw the diagram with pencil or pen.
It depends. It doesn't matter. You can, if you are confident that you will not make any mistake, you use the pen. But it's okay if you draw it with pencil also. It's a little bit. But whenever you are using pencil, make sure to make the diagram darker, or else it may be, when by the time it reaches the examiner, it might fade away. So it should be very darkened. so that it will remain as it is. It depends. It doesn't matter whether you are drawing it with a pen or pencil. If you are confident, you can draw it with pen also. But for these kind of answers, no, you have to use means a different what we can say different means different forms you have to use. Like one thing, one side should be shaded, one should be dotted, so that you can easily distinguish that this part is representing the promoter reason or this part is representing the UTR reason, like that. Okay, like what I mean to say is when you are presenting something that has multiple things within the same sequence or same location, then you should use different type of shading techniques to discriminate, so that it can be easily identifiable.
Okay, ma'am.
Okay, any other question from anyone?
हेलो मैम नमस्कार
हाँ नमस्कार को
मैम ହେଲା ଯୋଉ 3 ଦିନର ପିପିଟି ଏପର୍ଯ୍ୟନ୍ତ ଆମକୁ ତ ଗ୍ରୁପ ରେ ସେୟାର ହେଇନି ଆଉ
ନା ମୁଁ ତ ଗ୍ରୁପ ରେ ମତେ ଗୋଟେ ଗ୍ରୁପ ରେ ଆଡ କରିଛନ୍ତି WhatsApp ଗ୍ରୁପ ରେ, ମୁଁ ତ ସେଇ ଗ୍ରୁପ ରେ
ଗ୍ରୁପ ରେ
ଅଲରେଡି
ଅଛି ଗ୍ରୁପ ରେ ଅଛି
ଅଛି ମ୍ୟାମ୍ ପାଇଛି ମ୍ୟାମ୍ ଆଜିର ପିଡିଏଫ ଟା ବି ପଠେଇଦେବେ।
ଆଜିଟା ମୁଁ ପଠେଇନି, ଏବେ କ୍ଲାସ୍ ପରେ ସେଟା ପଠେଇବି, ବଟ୍ ବାକି ଆଇ ଥିଙ୍କ 3ଟା ଯାକ ଅପ୍ ଟୁ 3 କ୍ଲାसेସ୍ ମୁଁ ମେନସନ୍ କରିଦେଇଛି। ଆଉ ଇଫ୍ ୟୁ ନିଡ୍ ଏନି ଫର୍ଦର ହେଲ୍ପ ଅଲସୋ ତମେ ଗ୍ରୁପରେ ମୋତେ କ୍ୱେଶ୍ଚିନ୍ ଯେଉଁମାନେ କରିବ ଆଇ କ୍ୟାନ୍ ଆନ୍ସର୍, I have no problem in responding to your queries. Yeah. Over call it may not be possible, but if you have any kind of doubt, if you are writing to me through the WhatsApp, then I will definitely respond you. Okay, if you find any problem in understanding any concept or anything, you can simply write down your query or problem. I will get back to you. So that is all from
Thank
my
you,
side.
ma'am.
If you have any other question, you can ask, or we can sign out. And I wish you all the best for your government examination. And since you are connected through WhatsApp, whenever you have any question, like if you feel like discussing anything, you are welcome to do so. And Parvati, I hope you got your answer. The MPhil is no longer a valid degree. MPhil ebe uthe hi dechi government. Okay, even ebe government ta jo ama new education policy lagu hela pare, government actually promote korchi directly after BSc one can enroll into PhD. Okay. Like BSc ପରେ ଗୋଟେ ବର୍ଷ MSc କରିକି 4 year degree course କରିଦେଇଛି BSc ଟାକୁ. So after degree course, you can have 1 year of MSc and then you can directly enroll into PhD. So MPhil ଆଉ କୌଣସି utility ହିଁ ନାହିଁ. Okay, so if you don't have any other question, okay, thank you so much. I have tried my best. Actually, honestly, the problem is the time is very limited. And for genetics, particularly that mapping part and all, it's very difficult to teach online because a lot of means calculation and drawing are to be done. Means it has to be done by hand. So it's a little bit difficult. I don't know how far you understood that part, but I have tried my best, and I hope if you go through your material that time you can correlate this means teaching to understand it better. And as I told you, you are welcome to post your queries whenever you need. I will definitely respond to you. Okay. So thank you so much, and I wish you all the
Thank
best
you, ma'am. Thank you, ma'am.
thank you
government
ma'am
examination. Okay.
Thank you.
thank you ma'am
So ଯୋଉମାନେ
thank you
ଯଦି
so
added
much for
ନାହାନ୍ତି group ରେ,
so effective class.
Okay, thank you so much for your feedback. If there is anything I need to rectify, please let me know.
No
So,
ma'am
regarding the question answer, actually ଯଦି ମଝିରେ ବାରମ୍ବାର question କଲେ ଟିକେ flow ବି disturb ହେଇଯାଏ. Plus ମାନେ ମତେ ଲାଗେ କି maybe that might not be means suitable for others. Like ଯେମିତି ଏବେ କଣ ବହୁତ ଲୋକ left out କରିସାରିଲେଣି, so ତାଙ୍କର time ଆଉ waste ହଉନି. They don't have any query, so they can leave. Those who have the query, they can stay back and they can discuss. So in my view, that is better. କାରଣ ମଝିରେ କଲେ ସେମାନଙ୍କୁ ବସିବାକୁ ବି ପଡିବ, they have to wait, so they might think ଆମର time ଟା ଏଥିରେ waste ହେଇଯାଉଛି. So that is the reason I prefer to have a discussion at the end of the class. If you have any question in any slide also, we can go back to that slide and we can discuss also. There is no problem in that. Okay. anyway, so thank you so much. You all have been very patient and very cooperative. Actually, last year मतलब जी बहुत outsiders पिला थिले, but this time means most of ODIA students are there, so it was enjoyable to me as well. Okay. So thank you
Thank
all,
you.
Thank you, ma'am.
Thank you, ma'am.
and I wish you all the best once
Thank you,
again.
ma'am.
Okay.
Ma'am.
Thank you.